Hello, grade 9. Let's start our session for today. This is our chapter 3, lesson 1. It talks about the cell theory. This is page 66 in your book. We have one key concept, which is cells are the basic unit of life. The cells are the basic unit of life. And we have two main ideas. Number one, early studies led to the development of the cell theory. Number two, prokaryotic cells like a nucleus and most internal structures of the eukaryotic cells. This means that the prokaryotic cells are cells without a nucleus. Okay, let's first talk about the early studies that led to the discovery or the development of the cell theory. Almost all cells are too small to be seen without a microscope. Yes, we already know this. The early lenses was not powerful enough to show individual cells. So in the past, the scientists or people used the lenses to see the small things, but it was not powerful enough to show the individual cells. The invention of the compound microscope in the late 1500s led to the discovery of the cell. The invention of the compound microscope led to the discovery of the cell. A compound microscope composed of or contains two or more lenses. We already know that each lens or a lens is a part or a tool that is used to magnify things. So the total magnification power is the product of the magnifying power of each individual lens. The total magnification is generally much more powerful with a compound microscope than with a single microscope, for sure, because we are adding the magnifying power of each lens. So this will be much greater than the magnification lens of a single lens. Okay, then in 1665, the English scientist Robert Hooke used a three-lens compound microscope to examine a thin slice of cork. Cork is the tough outer layer or the tough outer bark of a species of oak tree. So he was discovering or examining plant cells. Hooke observed that cork is made of tiny hollow compartments, that he then gave them the name cells as we know them right now. The plant cells he observed were dead. He was looking only at cell walls and empty space. At the same time, Anton van Leeuwenhoek, he was a Dutch tradesman who was studying new methods for making lenses to examine cloth. He was a tradesman who was examining or uh, buying and selling clothes. Then he was making new methods for making lenses to examine his clothes, to be good to his trade. Then his single lens microscopes were much more powerful than Hooke's compound microscopes. His single lens microscopes, he was using microscopes with a single lens, but they were much more powerful than Hooke's microscopes, the compound microscopes. In 1674, Leeuwenhoek became one of the first people to describe living cells when he observed numerous single-celled organisms swimming in a drop of pond water. So he was examining the living cells. They are like the animal cells. These are the sketches of his observation, and he gave them the name animalcules. This is like molecules, but this is animalcules to refer that they are like the animals, but they are very small in size. Then as the people continued to improve the microscope, it became easier to use and capable of greater magnification. They increased the magnification power of the microscopes. This led people to examine even more organisms. The people observed a wide variety of cell shapes and cell divisions. Scientists began to ask important questions like, is all living matter made of cells? And where do cells come from? Then Shielding, this is a scientist named Shielding, used compound microscopes to study plant tissues and proposed that plants are made of cells. Okay, then another scientist named Schwann was struck by the structural similarities between plant cells and the animal cells he had been studying. Then Schwann concluded that all animals are made of cells. In 1839, Schwann published the first statement of the cell theory, concluding that all living things are made of cells and cell products. So this is the first statement in the cell theory published by Schwann in 1839. All living things are made of cells and cell products. This theory helped lay the groundwork for all the biological research that followed. This theory also had to be refined over the years as additional data led to new conclusions. Like what? For example, Schwann stated that the cells form spontaneously by free cell formation. The cells form spontaneously by free cell formation. Spontaneously means automatically, as later scientists studied the process of cell division, and they realized that this part of Schwann's idea was wrong. It wasn't true. Then in 1855, a scientist named Virchow reported that all cells come from pre-existing cells. All cells come from pre-existing cells. Then they concluded the three main principles of the cell theory, which are number one, all organisms are made of cells. Number two, all existing cells are produced by other living cells. Then, number three, the cell is the most basic unit of life. And this is the important part. These three lines are or they resemble the cell theory. Then we move to the second main point. Prokaryotic cells lack nucleus and the most internal structures of eukaryotic cells. Your body is made of trillions of cells of many different shapes, sizes, and functions. Yes, for sure, including long thin nerve cells that transmit sensory information, as well as the short, bulky skin cells that cover and protect the body. Despite this variety, they are very different from each other. The cells in your body share many characteristics with one another and with the cells that make up any other organism. Like what? In general, cells tend to be microscopic in size, very small in size, cannot be seen with your bare eyes or naked eyes, and they have similar building blocks. Cells are enclosed by a membrane that controls the movement of materials into and out of the cell. This is the cell membrane. Within this membrane, a cell is filled with cytoplasm. What's the cytoplasm? It's a jelly-like substance that contains dissolved molecular building blocks, such as proteins, nucleic acids, minerals, and ions. All these are dissolved in the cytoplasm. The cytoplasm contains organelles, the small organs of the cell. They are called organelles, which are structures specialized to perform particular processes within the cell. Most organelles are surrounded by a membrane. In many cells, the largest and the most visible organelle that you can see under the microscope is the nucleus, which stores the genetic information. Then we divided, or the scientists divided, cells can be separated into two broad categories based on their internal structures. Two, prokaryotic cells, and eukaryotic cells. Let's see what about them. The prokaryotic cells are like the one we can see right here. It is enclosed in a cell membrane and they have cytoplasm. This is bacterium. This picture is magnified 8,800 times. Can you imagine how small this is? Prokaryotic cells do not have a nucleus or other membrane-bound organelles. The cell's DNA is suspended in the cytoplasm. So the DNA of the prokaryotic cell is suspended in the cytoplasm. It is not in a nucleus. Most prokaryotes means the animals having prokaryotic cells. Most prokaryotes are microscopic single-celled organisms. So most of the organisms having prokaryotic cells are microscopic single-celled organisms. Then what about the eukaryotic cells? As the one we can see, it is larger, it have an obvious nucleus in the middle, and also it have other organelles. Eukaryotic cells have a nucleus and other membrane-bound organelles. The nucleus is the largest organelle and encloses the genetic material. The eukaryotes are organisms with eukaryotic cells. They may be multicellular or single-celled organisms. And this is the end of our lesson for today. I hope you understood. and see you inshallah in the next session. Thank you.